we have multiple honored guests today, including our visitor, Steve Kaczmarek from Michigan University. We also have our number of our prospective graduate students, I believe, who are on Zoom. So thank you again for joining us. Steve earned his bachelor's and PhD from Michigan State University, and then spent, I think, was 8 years and six. Sorry, I'm close. At Exxon before ended up at Western Michigan University. And this is really the really short story here. Is somehow I found myself working on the dolomite problem for some it's been great, but also like dolomite act. I can't believe it. And I was looking for someone to talk to, so I wrote an email to Steve. I mean, admittedly, it wasn't like 3-point font, fully justified, but the point is he was kind enough to answer. We struck up a conversation. Maybe we'll do a collaboration, and it ended up with him coming here to give a seminar today to share his wisdom with us about dolomite. And so thank you very much for joining us, and look forward to your seminar. Thank you, Steve.
Thanks for the introduction, Andy. Thanks to all for being here, virtually and in person. This is a talk that I've given a number of times before, and I've sort of it's evolved over time. And trying to get this out of the way so you can actually see the title.
Go one more. There's like a hide
Here.
something.
Hide floating meeting controls.
Yeah.
Sorry, where? There it is.
Yep.
Lovely, thank you. So the title of the talk called Poking Holes, Critical Reappraisal, Microbial Dolomitization, and I'm going to talk to you about some other useful applications of dolomite mineralogy. And what you're looking at here is a scanning electron microscope image of a crinoid that's been dolomitized, and I just thought it was very fitting because this crinoid, this central part of the crinoid is got a hole in it, and it fits with the title. This is work that I've been doing. Really, the story of this work starts. Hide media control. Sorry about that. This starts in 1999 as an undergraduate at Michigan State University, and if there's time, I'll tell you more about that. There we go. So what this says is it's the never-ending dolomite problem. And just as a quick primer, a dolomite is abundant in the rock record, but very rare in modern marine settings. It's, quote, difficult to synthesize at low temperatures in the lab, but most ancient dolomites have proxy data that suggests that the dolomites formed at low temperatures, so that's a bit confounding. And then there's a lot of different types of dolomite. The distribution is spatially and temporally kind of sporadic, and this ultimately leads to a lot of different proposed models of dolomitization. So let me just talk to you a little bit about what that word dolomitization means. I'm going to hide my floating controls again and see if that works. There's two ways to make dolomite in the rock record. Most of Phanerozoic dolomites are interpreted to result from what we call replacement, or this dolomitization. You start with a precursor limestone or carbonate sediment, usually calcium carbonate. You introduce some magnesium-rich fluids, and you end up with the mineral dolomite, shown by the reaction here. So what this does in terms of sort of the texture of the rock is we can follow two pathways during dolomitization. We can start with, this is showing a thin section of a grainy limestone, and it becomes dolomitized, and what we end up with is a crystalline dolomite. Or this limestone or carbonate sediment can be dolomitized, and it can look almost exactly like it started out. We call that fabric retentive dolomite. There's another type of dolomite in the rock record, that's dolomite cement, and that forms through direct precipitation from fluids, where you don't start off with a calcium carbonate precursor, but you have all the ingredients sort of coming from the fluid, and you end up with the same mineral here at the end. All right, so how do we study dolomitization? Well, we either go into the lab and we start with some well-controlled conditions and we observe the outcomes. Some of the controls we test are the temperature of the temperature of the fluids, the chemistry of the fluids, different reactants. ultimately, we're left with this question of how applicable to nature are our experiments. Typically, we do these at very high temperature compared to what we think dolomite forms in nature. The other approach is we go into the field and we observe the outcomes. We make lots of measurements and we use various proxies, which could be stable isotopes or trace elements, fluid inclusions. We're always left with the question of how accurate are interpretations if we're using a proxy record. And secondly, how certain are we that these proxy records are reflecting the initial conditions of dolomitization rather than a later diagenetic event where the rock has been recrystallized? And shown over on the right is a cartoon I found online that basically says, right, like a miracle occurs to make dolomite. And this is what drives everybody a little bit crazy and also takes people out of the research realm of dolomite because it is a bit confounding. What we know is that dolomitization is driven by a number of physicochemical parameters. And if you look at sort of the general equation here, what you know is you need a couple of key ingredients. You need a precursor calcium carbonate, shown here as like low magnesium calcite, and you need magnesium to get to the dolomite. In temperature, it's sort of thermodynamically, temperatures on the y-axis here, magnesium calcium ratio of the fluid shown on a log scale on the x-axis. What we see here is that there's sort of a sweet spot here where dolomite is stable at lower magnesium calcium ratios in the fluid. Calcite is the stable carbonate phase, and then at significantly higher magnesium calcium ratios of the fluid, magnesite, MgCO3 is the most stable phase. In terms of kinetics, these same parameters, the temperature and fluid magnesium calcium ratio, exert a strong control on the rate of dolomitization. Shown here is reaction time on the y-axis on the x-axis, and on the y-axis, the percent product, which is basically just the percent of dolomite in our experiments that we produce. And all this graph is designed to show is that when we use higher temperatures, the hotter colors represent higher temperature experiments, the rate of the reaction is faster. And so these are experiments conducted at 250 C, and then they step down to 160 C, and you can see that it takes a lot longer to make a given amount of dolomite in the reactions with lower temperatures. We see the same sort of effect in when we look at the magnesium calcium ratio of the fluid. Lower fluid magnesium calcium ratios result in slower dolomitization. So the more magnesium you have available, which is one of our key ingredients, the faster dolomitization proceeds. There's also a number of other factors that I'm not showing data for, but these are all proposed kinetic factors, stuff like the fluid rock ratio, the reactant surface area. Fine grain reactants tend to, or fine grain calcites tend to react faster than coarse grain, and so on. So ultimately, what I want to talk about is, sorry, I'm going to keep bouncing back. So
I think it might be because there's a chat, because there's chats and it keeps popping up.
It's not any new chat.
I know, and it keeps popping up because it's like, hello.
Oh, right.
So if
Okay.
you open the chat and then clear it, if it pops up again,
Okay.
that will fix it.
All right.
Just an idea.
So ultimately, what's all the fuss about with dolomite, right? And what we are commonly arguing over in dolomite is the who, the what, the where, the when, the why, and the how. And what that means is, how does dolomite form in nature, right? We can make it in the lab at high temperature. We see it in nature, and we think it forms at low temperature. And so we come up with these models, and basically these models are coupled chemical hydrological models. And what they're meant to do is explain a couple things. One is the source of that magnesium, and then how we get that magnesium to our calcium carbonates. And so the details here are not important, but what I want you to sort of get from this diagram is that we have a number of different models. And those models talk about the source of the magnesium and how we get those magnesium-rich fluids to our precursor calcite to make dolomite. And ultimately, every one of these models has a series of predictions that it makes, right? We have an explanation. We say the fluids are coming from this direction, or the fluids have these chemical properties, and we should see something result from that. we'll look at just one of these, which is called reflux dolomitization. It says basically we get highly evaporative fluids that sit on top of the carbonates that they get evaporated, they become dense. They sort of the chemical potential increases, and then they flow downward through the underlying calcite and they create dolomite. They do this dolomitization reaction. The one at the bottom here that I've added to Hans Machell's diagram is the what's called microbial dolomitization. And basically this model is what we're going to spend some time talking about today. Basically posits that microbes overcome kinetic barriers to dolomitization by changing the chemical conditions of the system such that dolomite can form more easily. So here's the microbial dolomite model in a nutshell. Microbes overcome these kinetic barriers to form dolomite at low temperature. It's of course plausible. Microbes are everywhere, and we know that microbes actually interact with their environment chemically and physically. The key evidence that is cited in the literature in support of this model is that low temperature laboratory experiments involving microbes are the only way to produce dolomite. Collectively, these papers are cited a couple thousand times, and many consider this microbial dolomite model to be the solution to our dolomite problem. It explains why we can't do the experiments at low temperature. So I'm going to talk a little bit more about the history of this model, but I need to back up a bit and tell you a little bit more about dolomite. And this kind of starts in the 1950s, where some researchers from nearby were attacking the problem of dolomitization through some experiments in the lab. And what they recognized is underlined in red here, that dolomites that were produced in higher temperature experiments achieved a stable 1-to-1 composition with respect to calcium and magnesium. So dolomite, the chemical formula is calcium, magnesium, and then 2 moles of carbonate. And so the dolomite they produced in these higher temperature experiments had pretty equal parts of calcium to magnesium. And the materials that they produced at lower temperatures were enriched in calcium relative to magnesium. There was another important distinction between the high temperature and low temperature experiments. The high temperature experiments had evidence of what they called cation ordering, that the calcium and the magnesium cations in the dolomite structure were in alternating layers, okay? Whereas the lower temperature experiments lacked that cation ordering. The magnesium and calcium were sort of distributed randomly throughout the crystal structure, okay? And so what they said is, we believe that the necessity of attaining an ordered arrangement of calcium and magnesium ions at rapid rates of crystallization is responsible for making dolomite at low temperature.
Quick clarifying question. We're talking about dolomitization, not cementation.
That's right. That's right. So going forward, pretty much everything I'm going to talk to you about involves creating dolomite through this replacement reaction. Thank you for the question. All right, so what does this look like? Well, here are two mineral models. One showing what they call protodolomite, showing basically a calcite structure where we have cation layers shown in red and blue that alternate with the cation, the carbonate anion layers shown in green here. So what you'll see is the cation layer and then carbonate, cations, carbonate, and so on, where the calcium and magnesium shown in red and blue are sort of randomly distributed. Dolomite, the ordered dolomite structure, is very similar in that we have cation and anion layers alternating, but in this case, the cation layers are predominantly all calcium in this case, and then magnesium, and then calcium, and then magnesium, and so on. So that's that ordered structure. These have different, these are two different minerals essentially, and that's attributed to their different cation ordering. So I like to think about dolomite and what they call protodolomite as polymorphs, right? We know the basic definition of a mineral from our intro geology classes. They're solid, they're naturally occurring, they're inorganic, they have well-defined chemical compositions, and all these are the same between what they call protodolomite and dolomite, but where they differ is in their regular crystalline structure, right? the pattern of the atoms within the mineral is different between dolomite and protodolomite. So they don't have the same structure. This is from a textbook just showing you to give you a better sense of what's going on within the lattice. This is a transmission electron microscope image showing sort of the atomic arrangement of these larger calcium ions is blown up in our kind of a cartoon here. You have the larger calcium ion layers, and then the cation, the carbonates, and then the smaller magnesium carbonate, calcium carbonate, and so on. And the arrangement of these things gives dolomite its characteristic rhombohedral structure. Now, importantly, the amount of magnesium to calcium in the crystal structure in dolomite can vary. And we call that stoichiometry. So the relative proportions of the calcium and magnesium can change. And then also the degree to which those calciums and the magnesiums alternate within the mineral structure can also change. And it's kind of lies on a spectrum. So how do we measure this? How do we really understand the degree to which the cation composition changes and the degree to which those calcium and magnesiums lie in their appropriate layers? Well, right now we use powder X-ray diffraction amongst other tools. And basically this is an approach to fingerprint a crystalline solid. So just like we have fingerprint patterns that are unique to different people, we have XRD fingerprint patterns that are unique to each mineral. And so we put this material into our diffractometer and we can measure the minerals that are present and their relative proportions by looking at the fingerprint. Essentially, what you're seeing here are reflections in XRD space, and these reflections are caused by constructive interference that occurs from successive and recurring planes within the crystal structure. And so different minerals, because they may have different compositions or different structures, say polymorphs like aragonite and calcite, they produce different reflections, and we read those reflections and interpret the mineralogy. So back to this work by Goldsmith and Graff with the dolomite and the protodolomite, they present these data. These are the X-ray patterns, and these lines here represent the recurring crystal plane within the crystal structure, and they're labeled according to their HKL indices. So these are just planes within the crystal that are measured. And what they found is that this particular pattern or fingerprint between dolomite, which I'm showing here, is different from the protodolomite and different from calcite. So here's the dolomite pattern I was just showing, and here's their protodolomite pattern. And I want to point out a couple important things. One is the main peaks here are all pretty much the same. There's a 012 peak, 104, and so on. But there's a couple key reflections that are missing. The 101, the 015, and the 021 that are missing in the protodolomite. These are what are called superstructure reflections or ordering reflections, and they're only present when the calcium and the magnesiums are distributed into their relative alternating cation positions. For comparison, if you look at low magnesium calcite, the pattern is very similar. We have some of the main peaks, but it is also missing the 101, the 015, and the 021 peaks. If we go into sort of the world of modern powder X-ray diffraction patterns, this is how we look at data these days. It's sort of degrees to theta on the x-axis and intensity or counts, X-ray counts on the y-axis, where each one of these little peaks, you may have heard people talk about X-ray diffraction peaks. These correspond to these different reflections seen in the strip diagram here. So same general patterns. You've got the 104, which is the most dominant peak or the strongest reflection here, and it's the highest peak in the modern pattern. Pointing out the 101, the 015, and the 021 peaks, those are ordering reflections. Those are only present when we have cation ordering. So back to where we started, right? So now you know what they mean when Graff and Goldsmith talk about dolomite produced at higher temperatures as the stable material, one-to-one composition, and ordered with respect to calcium and magnesium. What they're saying is we see the ordering reflections in our X-ray diffraction pattern. And this contrasts with the material that they make at low temperatures, which have a similar composition, slightly enriched in calcium, but they lack those ordering reflections, the 101, the 015, and the 021. So this is where we worked for decades. We knew we could produce this disordered magnesium calcium phase in low temperature experiments. We had only the ability to make ordered dolomite at high temperature. And then in 1980, this paper was published, another nearby or at least statewide institution, where Charles Manfield reports that uroliths in the bladder of a Dalmatian, a dog, were dolomite. And so he specifically talks about the X-ray diffractogram identified the mineral as dolomite and has a sharp 015 peak suggesting it's ordered dolomite. And assuming this Dalmatian wasn't, heated up beyond sort of, its body temperature, this is the lowest temperature record of making dolomite. It goes on to say, well, that, some possible explanations here are that the bacteria that are living in the urinary tract in the bladder of this dog may have altered the chemical conditions such that it could produce this ordered dolomite. So a possible insight emerges. And here's the data. This is a picture of the uroliths. And what I've got here is the original data. Sorry, I've sort of like, it was originally reported opposite to what I showed you before. So this image is inverted, but over top of it, I've put a dolomite standard in the red peaks here. And what I want to point out is that it has these ordering peaks, the 101, the 015, and the 021 ordering reflections. Importantly, all these peaks, their relative intensities and relative positions are spot on. And so this is, in fact, what mineralogists would look at this and say that's dolomite. That's an XRD pattern for ordered dolomite. So this really got everything kicked off for what's called the microbial dolomite model. And this is a paper that was published in Nature in 1995. I was an undergrad at the time. This was very exciting. I was writing a paper on dolomite for my strat class. And this paper came out. And it proposes microbial mediation as the possible mechanism for natural dolomite formation at low temperature. And what they say here is we describe experiments in which ferroin, or slightly iron-rich dolomite, with a fairly high degree of cation order was precipitated in the presence of sulfate-reducing bacteria. And these experiments were done at low temperature, 30 degrees C. Subsequent papers sort of build on this from the abstract of a paper published in Geology in 2008. Microbial experiments are the only proven approach to produce experimental dolomite under Earth surface conditions. And so you can see the stories building, right? This model is being accepted. It grows in stature. A paper 2008 says there's not much evidence to suggest dolomite as a biomineral, meaning like it forms predominantly through biological processes. And so we've got this model on our hands. This is a widely cited model. Like I told you, the papers are cited thousands of times. And again, the key evidence that is used in support of this model is that these low temperature laboratory experiments have produced ordered dolomite, and we've not been able to do this before. So what I'd like to do over the series of the next few slides is to look at some of the evidence that was published in these papers together with you and point out some of the problems. As a refresher, right, dolomite is ordered. There's no single dolomite peak. We need sort of all these peaks, and we need them in their appropriate positions and their appropriate relative intensities sorry about this. This is just a list of all the peaks that one expects to see. These are the same HKL indices that are labeled here. And I've shown this diagram before. So I want you to pay particular close attention to the positions, the presence or absence, and the relative intensities of these key peaks that we call the ordering peaks. So here's a study from 2013. You can see the claim is in the figure caption: dolomite precipitated in association with the cell wall of the methanogenic bacterium. And they say the dolomite main peak is in the right spot. And so here's the data, right? And they sort of say, here's the main dolomite peak. That's that really tall 104 position. And they have some images here. They have some elemental data showing that, in fact, this stuff does have magnesium and it's got calcium in there. But this is their X-ray diffraction data, right? So this is where the main dolomite peak is. This is where the ordering peaks would be. We can't really evaluate the 021 peak. This is where that 015 peak should be, and here's where the other one would be. The fact that this peak is so small suggests that we would never be able to see those minor ordering peaks anyway. So here we have a case where the amount of material that was made is so small that we really can't use this tool to evaluate. We can't evaluate the claim. Here's a blow up of the X-ray diffraction pattern, and that's their dolomite peak right there. Here's a study from Geology 2005. The claim here, note this strong presence of ordering reflection peaks indicating the mineral produced at higher temperatures, 40-45, is fully ordered dolomite. What I've got here is the data that was presented in this paper, and the red curve here is the dolomite standard. So what I want you to see is that this 104 peak matches up with pretty well with a peak that they have in their experimental results. But when we look for these ordering peaks, like the 101, the data were not collected down that far, so we can't evaluate that one. The main ordering peak, the 015 here, is missing, clearly missing in their higher temperature experiments and all their experiments really. And then the 021 peak is also missing as well. And so what we think here is that they've got the same kind of phase that Goldsmith and Graff had, which was the proto-dolomite, a calcium magnesium carbonate, but it lacks cation ordering. Here is another paper. This is the slide that I typically get a few interesting reactions, raised eyebrows. This is from a Geological Society of London special publication. This is taken from their figure caption. It says the main dolomite peak is present at 30.99 degrees 2 theta, and a number of subsidiary peaks can also be identified, including weak ordering peaks at 37.4 degrees and 41.2, confirming the mineral's dolomite. Remaining peaks are from aragonite grains in the vial. So these experiments, they start off with some aragonite, they add some bacteria and some magnesium-rich fluids, and they cook them essentially. And they analyze them. And so here is their published pattern. And here is our dolomite standard. So the 104 peak lines up pretty well. Again, we can't evaluate this lower ordering peak. Here's where the 015 ordering peak should be. It's not really there. And the 021 peak's not really there. But here's where it gets kind of almost comical. If you look at aragonite, this is the aragonite pattern. So this is what they started with in their experiments, and it's almost a laydown match for what they ended the experiments with. So, this is just a case of mistaken identity. They were looking for peaks in the 104 region around 31. They were looking for peaks in the general vicinity of 43.7 and 35.3, and they saw some peaks, but they just simply mislabeled them, and they forgot to look at their aragonite standard that they started with. This is from that original Nature paper. This one's a little bit more complicated. But again, the black is the data shown or presented, and the red peaks here are the dolomite standard that's standardized to their pattern. This is where we look for the 101 peak down here. You can kind of see it's pretty noisy data, so really hard to say convincingly if that's there. They're missing altogether the 006 peak that should be sitting in this position. Here's our 0.15. It really should be around 35.3, where that little red dot is by the laser pointer cursor. This is where they marked it, and notice it's in a lot higher, it's got a lot higher intensity than we would expect. So what this would suggest is this is even more ordered than ordered dolomite, which can't really happen. And then we've got the 113 peak here. It's pretty noisy again. And then our 021 ordering peak is sort of missing. And then these peaks up here around 50, which is the 018 and the 116. You've got one of them, but you don't have the other one, and the intensities are sort of backwards. So there's just a lot of problems with this pattern. And probably what it reflects is they've got a lot of different stuff in here. They've got quartz, they've probably got aragonite, they've got probably hydromagnesite. So there's just so much stuff here, you can't even really evaluate the data. But here's the claim: the presence of the ordering reflection, the 015, indicates that the mineral is fully ordered dolomite. And like I said, this peak is too high actually to be the 015. It doesn't work that way. Here's the last example I'm going to show you. This is from a series of experiments. This comes from the geology paper produced in 2008. Where they said the microbial experiments are the only proven approach to produce experimental dolomite under Earth surface conditions. And you'll see these are the data from a series of experiments conducted at 35 and 25 degrees, where they use different bacterial cultures. The dolomite standard is shown down here, and they also identified some hydromagnesite, which is down here. And sort of for ease of viewing, I've highlighted the standards in different colors. And then I've plopped those over top of their patterns. And you can see this gets really complicated really quick, hard to see this stuff. So we'll just take a look at one, and I've blown it up. And essentially what you're seeing here is a big mess. It's really hard to evaluate this. But a couple things worth pointing out. Some of these peaks that they have labeled as dolomite are just too high. They're higher than you would expect for perfectly ordered dolomite. This is where the story gets kind of really interesting, and I don't want to bore you with all these little wiggles, but a subsequent study that was published in 2012, American Mineralogist, claims that the authors of that 2008 study distorted their standard patterns to prove that the strongest peak is from dolomite. So here is some evidence that those patterns were actually stretched and sort of distorted in different directions to produce a pattern that more resembled dolomite than the original data did. So it gets a little icky there. So let's backtrack and remember where this began, right? So these experiments conducted by folks in Illinois said, at high temperature, you make ordered dolomite. At low temperature, you make a disordered phase. And most of the data published in these low temperature microbial experiments look like they don't have the right ordering structure either, and are a mixture of hydromagnesite, aragonite, what we call protodolomite, or very high magnesium calcite. More recently, that same paper showed that you could make this phase, this disordered, what they call disordered dolomite phase, at low temperature, just using some carbohydrates, some polysaccharides. You don't need any active microbial metabolism going on either. And so the model here, the microbial dolomite model, that we're somehow using microbes to change the kinetics and change the chemical system, is kind of on a little bit shaky footing now, because all the experiments haven't produced this ordered dolomite, and subsequent experiments show that it's pretty easy to make this disordered phase that we see in the experiments. So up to this point, what I want you to know is that these published XRD patterns are pretty inconclusive for a variety of reasons. They match patterns of other common and carbonate minerals like aragonite. They're questionable, or they lack the dolomite ordering reflections, and some of them have been altered or fabricated. And then secondly, I'm not going to go into detail, but I also want you to know that the published microbial products kind of look like this, these dumbbell microbial shapes. They almost all look like cements, material that's precipitated out of solution, Whereas the vast majority of dolomite in the rock record looks like these little rhombohedral crystals, crystalline dolomite, or does this replacement, we call it mimetic replacement. It retains the shape of the limestone. So in terms of mineralogy, it's not like dolomite we find in the rock record, and petrographically, it's very dissimilar to the dolomite we see in the rock record. So the conclusion here is there's really no compelling evidence that the materials produced in these low temperature microbial experiments are dolomite as claimed, and this kind of undermines the model for me. So now I'd like to change directions and tell you a little bit more about like how we apply dolomite mineralogy to other questions. Yes.
Just one clarifying question. I don't know a lot about XRD, but how much of this mismatch could be due to changes in technology over time? In terms of the ability to get the peaks where you want them to be?
Yeah, the technology is not evolved all that much, honestly. I mean, the X-ray diffraction work that was done in the 50s is really high quality stuff. The general machine that we use, they add bells and whistles to it, but it's doing the same thing effectively. Unlike geochemical proxies where you see that uncertainty value through time reducing, we don't have that X-ray diffraction. Good question. So here's what what we've learned by studying a lot of dolomites. We know that the degree of cation ordering and the composition vary widely. So what we do is we go into these patterns, okay, these X-ray diffraction patterns, and we measure two things. One, we measure the position of this 104 peak, and it shifts based on the amount of calcium and magnesium in the mineral structure. Magnesium is a smaller cation than calcium, and so the more calcium you have relative to magnesium will push this the position of this 104 peak, and actually the whole pattern, and generally it shifts it to the left. Okay, there's an inverse relationship between the 2 theta angle and the D spacing, the interplanar spacing within the mineral. So more larger calcium ions means a smaller 2 theta angle, and it pushes the whole pattern to the left. More magnesium pushes the pattern to the right. So we measure that. We also measure the ratio of this 0.15 ordering peak to another peak in the pattern, the 110, which is not an ordering peak. 110 is there if it's calcite or if it's dolomite. And so as this ratio increases towards 1, the inference is that we're getting more and more ordering. So when we go into nature and we measure lots of dolomites, like over 1,600 dolomites, what we see is that the stoichiometry, the amount of magnesium in the dolomite, varies from about 39 mole% all the way up to about 54. We also see a highly variable ratio of that 0.15 to 110. So the cation ordering goes from basically 0 in these natural rocks all the way up to over 1. So really what we have is we have a spectrum of minerals that we just throw into this single name we call dolomite. So who cares? Right? How do we use this information? How do we use the knowledge that dolomites vary in their composition and in their degree of cation ordering? So one, we need to know what the controls are. What causes more magnesium or more calcium to be in this dolomite? Well, there we went into the lab and we looked at a number of key parameters. One is when you change the magnesium calcium ratio in solution, we show a positive correlation between that and the amount of magnesium in the dolomite. So pretty simple. More magnesium in your fluids, we showed faster rates, but also the material you produce has more magnesium in it. Pretty strong relationship. Same goes for temperature. Increase the temperature, you increase the rate, and you increase the amount of magnesium. So you make a dolomite here at 160 with about 36 mole% magnesium in it, and you make one at 250 and it's about 46. So pretty big difference. You can also change the salinity of those fluids, where the more saline fluids correspond to a higher degree of magnesium in there. And then other work has shown increase the amount of carbohydrates or polysaccharides in the fluid, and you get more a higher degree of stoichiometry. So some key parameters that vary in nature, right? Natural fluids vary in their temperatures, their chemical composition, salinity, and the amount of organic matter that's in them. And this all has a control on the dolomite stoichiometry. What about cation ordering? Well, we know that cation ordering develops over time, and that it's linked to temperature and fluid chemistry like our stoichiometry, but it's also related to some kinetics and defect microstructure, like the interior microstructure of the mineral, and growth mechanisms. So what you're looking at here is a plot that shows the cation ordering. So again, the ratio, the 0.15 to the 110, as a function of reaction time. So we start with some calcite or aragonite, we mix up some magnesium-rich fluids, we put these in little autoclaves, little vessels, and we put them in the oven. And then periodically we take this material out, we filter off the fluids, and we grind up the solids, do X-ray diffraction work, and we plot these. And what you'll see here is early in the reaction, we start off with really disordered materials, and over time, these materials become progressively ordered. Now, not only do they get progressively ordered, but they also change their fundamental, like the surface of the crystals changes shape as well. Where you have disordered phases, I had this really bumpy, we call it mound microstructure. And later on in the reaction, when you have like well-ordered dolomite, you have a very flat layered structure. And so we've used this data to infer different growth mechanisms. The material here is growing sort of very sporadically, materials adding to the crystal surface in kind of an unruly way. Whereas later on, it's much more deliberate. It's growing by these more of a, we'll call like a layer growth mechanism. All right, so how do we apply this knowledge? Well, in conjunction with other petrological data, we have done a lot of different things. So I've shown you that we've used this to evaluate claims of laboratory dolomites. We also do this to evaluate claims of modern dolomites. We can use these data, the stoichiometry and cation ordering data, to look at various dolomitization models. I'm going to show you the example of evaporative reflux, that model where we have those evaporative fluids that are fluxing down through our carbonates and dolomitizing them. Show you two examples. One's a large scale top-down reflux model, and the other is what's called a cycle scale evaporative reflux. We've used this to look at dolomitization in lacustrine settings. We use these to look at late stage fault control dolomitization in deeply buried strata and in deep basins. We've been applying this mineralogical fingerprint to map out sedimentation and circulation patterns in detrital sediments in the Arctic. We use these data to evaluate the texture and geochemical resetting that's involved in recrystallization. And then I'll talk to you just real briefly about a project that we're working right now where we're evaluating mass-dependent stable isotope fractionation in these carbonates. So here's the general model of evaporative reflux, or what's called seepage reflux. You have a restricted environment here. This is like a carbonate platform, for instance. It's a rimmed platform. So we've got restricted lagoon back here. You've got evaporation. These fluids become dense and saline, and they flux downward through the underlying carbonates and they dolomitize it. So this is the reaction. We see an association between evaporites and dolomites in the rock record. And so this model is really common. It's probably the most popular dolomitization model beside the microbial dolomite model at the moment. The reflux model predicts some stuff. These are some reactive transport models done by some former colleagues of mine at ExxonMobil Upstream Research. And basically what they did is they used these coupled chemical hydrological models to test reflux dolomitization. So they injected a brine into this 2D model, and they showed how this brine fluxes downward through this underlying sediments and dolomitizes. So the red is like high degree of dolomite, and blue is the limestone. So you can see after 0.2 million years, there's sort of this gradient between fully dolomitized to partially dolomitized into the undolomitized limestone. And if you let this model run long enough, what happens is you just dolomitize this whole section. And this has been used as sort of supporting evidence for this model. one of the outcomes of this model is that distance from where the brines enter into the limestone, because of that reaction that you're sequestering magnesium from the fluids into the mineral and then liberating some calcium back into the fluid from the calcite, what happens is the magnesium and calcium concentrations change, right, in the fluid. So down gradient, what's expected is that as we're dolomitizing, we're losing magnesium and we're picking up calcium. And so what that does effectively is for every parcel of water down gradient, the magnesium calcium ratio of those fluids goes down and down and down. And so it's losing dolomitizing power along that gradient. The cool thing is that that's one of our key chemical parameters that changes dolomite stoichiometry. And so we can look at spatial changes in dolomite stoichiometry to test this model. And so let's look at this example from little Qatar here. We're looking at some Paleocene Eocene rocks called the Umaraduma Formation. Umaraduma Formation is basically a big, thick stack of dolomites that sits below some gypsum. And the model here is that conditions got really shallow, really evaporative. We started precipitating gypsum, which sucks calcium and sulfate out of the water. And you had these really magnesium-rich fluids that were dense and they fluxed downward through the underlying limestones and they dolomitized it. This model basically predicts, right, a downward decrease in dolomite abundance, a downward decrease in dolomite stoichiometry because those fluids are running out of gas, the magnesium calcium ratio is reducing. We might expect heavy delta O18 of those dolomites. We might expect elevated trace elements in the dolomites because of these evaporative fluids. And we might expect some relationship between those two mineral phases because one was driven by the other. So let's take a look. So here is just a mineralogy section. Essentially, it's all dolomitized, so we can't use the abundance of dolomite to say much. The magnesium, the stoichiometry of the dolomite at the top is around 49.5, and then it actually increases here through the middle of the section and then decreases. This is not really consistent with our predicted value of being highest near the top where those fluids were coming from and decreasing downward. It's in terms of the delta O18 values, this is where we would, this dotted line is sort of the Eocene delta O18 of seawater. So we might predict the dolomites to be a little bit heavier, but in fact, they're all lighter than that. So this doesn't jive with these warm evaporative fluids either. And then in terms of trace elements, strontium and sodium, we would expect values for marine dolomites that are sort of in the range that we observe, and evaporitic dolomites tend to have much higher values than we observe. So our stoichiometry pattern, our stable isotopes, and our trace element concentrations don't really fit with this story of top-down evaporative reflux of these fluids. It's also inconsistent with the timing because everywhere we look petrographically, what we see is these little dolomite crystals here. They always predate or are included in the gypsum. So the gypsum that formed there is not forming and then causing the fluids to be magnesium rich, driving dolomitization, but it's the sort of the opposite. We're forming dolomite and then the gypsum comes in and cements everything together. Another example here, this is some Cretaceous rocks called the Upper Glen Rose Formation. They appear in the Comanche platform here in the Albion. And what is hypothesized for these rocks is it's a series of depositional cycles like parasequences. And there's two main models that were talked about here. One is what's called per cycle reflux, where each one of these cycles got evaporative and then fluids flux down. So cycle one, it shallows upward, you get evaporative fluids, those flux downward. Then you get another depositional cycle, the fluids flux downward and so on. And then another model where actually each successive cycle, those fluids actually bleed down into the underlying unit. And so the main idea here is that we have the cyclical sedimentation, and we're trying to figure out how these rocks were dolomitized. So we can apply a similar test like we did in the previous example. So in model one, this is fun. Model 1, top-down fluid from each cycle cap as shown in the diagram here. We might expect downward decreasing dolomite abundance in each cycle as those fluids run out of gas. We might expect decreasing stoichiometry in each cycle because, again, the magnesium calcium ratio is decreasing. And because this is all driven from evaporative fluids, we might expect elevated delta O18 values. In model 2, again, these patterns would be blurred and actually might show more dolomite at the bottom because each successive cycle is benefiting from the dolomitizing powers of the fluids from the overlying cycle. So cycle 1 is getting hit with one generation of fluids and then getting hit with the second generation and also getting hit with the third generation. And so these are sort of the patterns we would expect, right? For model 1, you get these downward changing patterns, and in model 2, kind of complicated pattern. And here's what we see. Instead of these top-down patterns, what we see, in fact, is dolomite abundance and dolomite stoichiometry that are very cyclical, such that the observed patterns don't really match those predicted for either model 1 or model 2. What we see is dolomite abundance, the stoichiometry, and the delta O18 all track with depositional facies. So as the facies become deeper and then shallower, what we see is dolomite abundance decreases and then increases, stoichiometry decreases and then increases, and our delta O18 values decrease and increase. And so the more dolomite we have, the more stoichiometric it is. And the heavier oxygen isotopes we have, and then I'm not showing here, but those are our highest delta 47 crystallization temperatures. They also have the most, the highest concentrations of trace elements. And so what this is telling us really collectively is that these dolomites are forming very, very early as the fluids are changing. So as our fluids go from shallow to deep, these values are decreasing because the fluids are getting less powerful. They might be lower temperature and not as saline. Those 3 key things that drive dolomite stoichiometry, right? Higher magnesium calcium ratios, higher salinity, and higher temperature. But we're seeing deeper facies and lower stoichiometry. So in this model, we think we can rule out model 1 and model 2 because we just don't see those patterns. But in fact, we see a very early dolomitization that's following the facies. And then this is the last thing I just want to show really briefly. This is a current project we've got going where we're trying to evaluate the controls on stable isotope fractionation. Essentially, we're trying to answer the question, do the various phases that we've recognized in both the rock record and in our experimental dolomites, do they fractionate fractionate stable isotopes differently? And so what you've got here is a plot showing time, and these are experimental results versus the relative abundance of the solids. And so in this set of experiments, we start with aragonite, and you can see we start with 100% aragonite, and over the course of the first few hours, that aragonite starts to dissolve. And when it starts to dissolve, what we produce is what we call this very high magnesium calcite, protodolomite, disordered dolomite. So we form that, and then that starts to dissolve at the expense of this poorly ordered dolomite that starts forming. And then that starts to dissolve at the expense of this well-ordered dolomite. And so this reaction, this kind of equation shown up here says, okay, we start out with our aragonite, we add magnesium, and we get a very high mag calcite, and then we make a poorly ordered dolomite, and then we get a well-ordered dolomite. And again, what we're trying to figure out is, do each one of these phases fractionate stable isotopes differently? And our preliminary data suggests it does. And the theory behind this is, again, back to this, the growth mechanisms. If this poorly ordered dolomite, this very high magnesium calcite are growing by one growth mechanism, they may be fractionating stable isotopes differently than this well-ordered dolomite that's growing by a very different crystal growth mechanism. All right, so key takeaways, walk out of here and have learned nothing but this, that dolomite stoichiometry and cation order are important mineralogical attributes. People don't measure them. I review a lot of papers where people talk about dolomite, they talk about the chemistry, there's no XRD data, there's no mineralogical data. This is sad because they're pretty easy to measure. It's easy to grind up a sample. Most geological or science departments have a powder X-ray diffractometer in the department. And then hopefully I've convinced you that there's a lot of potential applications for this. So here's just an X-ray diffraction pattern from dolomite. And it's just cool for me to tell you that we just use these little peaks here to learn all sorts of interesting stuff about nature. And I'll leave off with that. I'll thank you for your attention. This is a group photo that we recently took on campus. These are my 3 PhD students that I currently have. At one point I had 9. The group has shrunk quite a bit, but that leaves a lot more time for taking care of my 10-year-old at home. If you'd like more information, website shown there, and I'll take any questions that you have. Yeah, in the back.
Thank you. That was a fascinating talk, and I have a lot of things that I probably need to go think about more deeply for a while before asking them. So I will follow up at social hour.
Great.
The one thing I want to push on a little bit is sort of this question of recrystallization versus crystallization. Because in initially those Vasconcelos papers that you're citing, those microbial dumbbells that they're observing are presumed and hypothesized to be a crystallization phase as opposed to a recrystallization phase.
That's right.
And you rebutted that in the discussion of the microbial dolomitization model in favor of something that might be more chemically driven, albeit chemical derivatives from microbes like cellulose. But then in the stratigraphic data you just presented, we're looking at something that is very early. Why is it not just primary?
Great question. So one of the things that I sort of took out of this talk is sort of the question is what they're making in those experiments, some sort of like real initial phase that we're just not seeing in the rock record, right? And I've talked to Chris and Judy and all the people that have done this work and said, you know, what we really need to do is we need to take the stuff you're making in the lab and then give it to me and let me recrystallize it, right? Like this is probably the answer. But I don't think they've run those experiments long enough. That's first thing. And then the second thing is the claims as is like this is the solution to the problem without sort of actually demonstrating it. To your question about the stratigraphic stuff, we have a lot of petrographic evidence that the Dolomites are a secondary phase, right? We have preserved limestone in those sections. So in the shallowest facies, we have the most replacement Dolomite. In the shallowest, we have forams and like calcite mud. So we know that this depositional system is actually a pretty typical carbonate depositional system that's been dolomitized. Sorry?
A pretty typical carbonate depositional system from the Phanerozoic.
Yes, from the Phanerozoic.
That's the
Yeah,
big old asterisk on everything.
it certainly is. Yes.
In Michigan, you have in the Marquette supergroup, the Precambrian cone of dolomite,
We do.
and it shows petrological evidence of evaporative setting. And I'm wondering if you have any recent thoughts about primary versus evaporative reflex versus
Yeah, so I would say 99.99% of all my work is done in the Phanerozoic. The Kona dolomite is one of those dots on that N equals 1600 samples there. It's very well ordered and very stoichiometric, which commonly suggests to us it's probably been recrystallized. The Precambrian stuff, I tend to just sort of plead the fifth on it because I haven't looked at enough. I'm aware of the debate between the primary versus a replacement, but I'm not educated enough in my own work to really comment on it. I appreciate the question though.
At least to the people in the room, the closing for the grad student thing is supposed to start at 11, but we'll push it to 11:15. So if we want to take like one more question, we probably can do that, or a couple more. Yeah, you had a question.
Yeah, I mean, it was basically a lot of what Maggie was talking about, but I mean, there was a lot there. I wish you would come like a month ago. Dissertation writing wise would have been helpful, but this is super interesting. I guess the $1 million question that I have is like, if you have a dolomite, like what's the best way to tell the difference between cementation or dolomitization, right? Like primary precipitation, like based on your methods. Any?
Yeah, that's, the mineralogy is not going to be super useful there. It's mostly petrographic evidence.
Right,
You know, I'll share a paper with you later that people have argued that a lot of what we call replacement dolomite is actually like cementation. And I think we get a little bit into semantics when we start really talking about that. Where I started my career in research at an oil company, the way we drew the distinction was, is it filling up like pore space in between the crystals? Then we call it a cement. Are they like dolomite ROMs, or have they replaced allochems or grains, right? Then it's replacement.
but can't you get primary precipitation of a dolomite?
Sure, you can. It's just
That's not a cement, I guess.
yes, and that's where in the Precambrian, I
Yeah.
think that's the debate, right? That people are using these dolomites and proxy measurements from them and saying this is reflecting fluids during the Precambrian, right? But in the Phanerozoic, the overwhelming majority of dolomites retain some evidence of the limestone precursor. So that's a
Yeah.
lot easier problem to solve.
Right, and so can you apply XRD, I guess, to define that versus the proto? Because I don't know, we were just, I mean, all the information is like swirling in my head, but like the proto dolomite versus order dolomite and the time that it takes and like the fluid composition and all that, like making that like primary precipitation dolomite, like would you be able to tell using XRD? Like, because it could be anywhere on this, it could be proto, it could be ordered, and it still could be primary, right?
Yes, so I think what you're getting at is a fundamental question that I have too, that do all like dolomites, whether they're cements or replacement, do they all start out disordered and then they're
Right.
recrystallized? And I think the answer is probably yes. But I don't have the data to prove that yet. But I do know that when we go out into more modern settings, right, and you look at, I can show you. Here's a study by 2010 study. These are microbial mats that they say there's dolomite in them, and kind of here's their X-ray diffraction pattern. And of course they're complicated because they've got what they call anorthite, there's calcite, there's halite, there's quartz, there's all sorts of stuff in here. I don't know what this is. Here's the standard put over top of it. And again, like there's a lot of mess in here, even with what they call modern dolomites. We've reanalyzed stuff from Andros Island that people said was modern dolomite. And the evidence isn't very good that they're ordered. So I think a lot of this stuff that starts off disordered and then sort of ripens, for lack of a better term.
Cool.
But it's still, depending on where it ripens, I think it can retain some original geochemical signatures. And the example from the Upper Glen Rose Formation, the Cretaceous dolomites, where you see those cycles, you see mineralogical, geochemical, elemental, isotopic, delta 47, trace element, all this stuff is matching the facies. To me, is screaming that you've got kind of a primary record or a very early record. So maybe those dolomites are capturing the fluids at the time of deposition. Right? That's our holy grail. Like with all proxy, we're constantly looking for minerals that retain the original signature. And
so you're like, it doesn't even matter if it was primarily precipitated or not, if we're still retaining.
That's
OG
right.
Stuff.
If I replaced, let's say I replace a foran with dolomite in marine waters, that's just as good as if I grew dolomite cement in marine waters, right? It's locked in that signature.
Yeah. Cool. We'll talk later.
Yeah,
Have more.
look forward to it.
Thank you, Steve. That was awesome.
Thanks, everybody.
Yeah.